Good morning and welcome to the GW Epilepsy Grand Round. Today I'm delighted to introduce Dr. Jack Parent from University of Michigan. Dr. Parent is the William J. Herdman Professor of Neurology at Michigan Medicine. He earned his undergraduate degree in human biology from Stanford with distinction and his medical degree from Yale University. And then he went to UCSF for his neurology residency and clinical neurophysiology and epilepsy fellowship. In 2000, he joined the Department of Neurology at University of Michigan, where he currently serves as co-director of the Epilepsy Division and director of the Human Stem Cell and Gene Editing Core. Dr. Parent's research focuses on understanding mechanisms of genetic and acquired epilepsies using stem cell and developmental neurobiology approaches. He has pioneered the study of adult neurogenesis in experimental temporal lobe epilepsy and the use of human-induced pluripotent stem cell models of genetic epilepsies. Working with Dr. Laurie Isom, he has applied mouse and induced pluripotent stem cell models to understand mechanisms of seizures and SUDEP, sudden unexpected death in epilepsy, in genetic ion channel epilepsies. His lab has recently applied in vivo rodent gene editing techniques to advance understanding of non-ion channel genetic epilepsies. Dr. Parent has earned a lot of awards for his research, including the Dreyfus-Penry Epilepsy Award from the American Epilepsy Society, the Grass Foundation Award in Neuroscience from the ANA, American Neurological Association, and the Basic Science Research Award from the AES. And he recently served as secretary of the ANA and on the board of the AES, and he has founded and now co-chairs the Scientific Advisory Board of the Dravet Syndrome Foundation. He's also chief editor of Epilepsy Currents. Please join me in welcoming Dr. Parent to GW.
Thank you, Mohammed. I want to thank you for the invitation and the GW Epilepsy Group. Can everybody hear me okay? So I'm going to tell you about our work applying stem cell biology approaches to understand genetic epilepsies. For many years, over 20 years, I've been studying acquired epilepsy, specifically temporal lobe epilepsy. And our group and other labs have made, tremendous advances in understanding potential epileptogenic mechanisms. But with these acquired epilepsies, the insult to the brain that leads to the spontaneous recurrent seizures causes hundreds, thousands of changes in the brain. And trying to understand the critical few changes that lead to seizures is really difficult. So about 10 years ago, my lab, part of my lab, changed focus to study monogenic epilepsies with the idea if you start with a known gene mutation, perhaps we can understand all of the mechanisms that give rise to changes in brain function, in neuronal function, brain networks that lead to seizures. And there's been a lot of progress in genetic epilepsies, but we still haven't solved any of them. So we need better models. These are my disclosures. They're not applicable to today's talk. Many of you have seen this slide from Ingo Helbig. And basically, he divides genetic discovery in epilepsy into two eras, the channelopathy era in the 90s, where you'd take large pedigrees and do gene mapping and come up with epilepsy genes, and most of these are ion channels. But then with next-gen sequencing, there's been an explosion in the identification of epilepsy genes. We're well over 100 genes that cause different epilepsies. And they're not just ion channels, they affect synaptic function, cell metabolism, gene expression, other cell signaling features. And that's really given rise to the potential to study lots of different causes of epilepsy, but we need better models to understand the mechanisms of these genetic epilepsies, and hopefully this will lead to precision therapies for these patients. And there's been a lot of progress, zebrafish genetic epilepsy models, different new rodent models. Today I'm going to focus on human pluripotent stem cell models. These genes that are circled are just a subset of the genes that we study in my lab. The problem is every student or postdoc wants their own gene to study, so that ends up being a lot. But today I'm going to talk about the genes circled in red, some of them only briefly and some of them in more depth. The last gene I'm going to talk about actually was work I did in Ben Novich's lab with his group while on sabbatical at UCLA, which was nice to spend last winter in LA instead of Ann Arbor. So we're focused on human pluripotent stem cells, which are two types of cells, human embryonic stem cells and induced pluripotent stem cells. And induced pluripotent stem cells, or iPSCs, were discovered by Shinya Yamanaka in 2006 in mouse, and then his lab and another lab the next year in 2007 in humans. And the importance of the discovery was such that he, within 6 years, was sharing the Nobel Prize for this. So it was really tremendous. And the basic idea is you take a mature cell, like a fibroblast obtained from a patient skin biopsy, and you force the expression of specific transcription factors, and that reprograms the cells into these pluripotent stem cells that can, in theory, make any cell type of the body. And if we're studying epilepsy, we want to differentiate them into cortical neurons and then look for a phenotype in the patient cells versus control cells. So why focus on human cells? Why not just study mice? Well, there are a lot of important differences between mouse and human brain, especially with regard to development. All of you know the size and gyral pattern difference between mouse and human. Mice are lissencephalic. But in addition to that, the stem cell niche is very different. So when our brain develops, the stem cells are in the ventricular zone. They're radioglia. They give rise to progenitors in the subventricular zone. And compared to mouse, the primate, including humans, subventricular zone is markedly expanded. And they have a cell type called the outer or basal radioglia that are not really present in rodents. And these subventricular cells are thought to give rise to the superficial cortical layers and lead to expansion of the primate cortex. So if you want to study these things, you can't study them in mouse, you have to study them in human models. In addition, neurogenesis in the brain occurs over 6 days in mice, and it's well over 3 months in humans. So it's a very different process. So in addition to studying mice, we focus on the human cell models, and we've done a lot with 2D human pluripotent stem cell models. So basically they contain the patient's genetic background, including the gene mutation that we're interested in. You can make a nearly unlimited supply and differentiate them into many different cell types. You can do CRISPR gene editing on them, which is important. So if you have a line with a mutation, you can use gene editing to correct it and give you an isogenic control that differs only by the presence or absence of the mutation. Or if you don't have access to a patient, you can use CRISPR to put in a mutation and make a virtual patient. So you have the original control line and a new virtual patient line to compare. And then I'm going to talk more about the cerebral organoids, growing them in 3D culture. But for 2D, basically, we take patient and control cells, differentiate them into excitatory neurons, inhibitory neurons, make mixed cultures to model brain networks. We can look at gene and protein expression, single-cell electrophysiology, and multi-electrode array recordings where we plate them on the electrodes and can record longitudinally over time. We can also do the same thing with calcium imaging to look at activity. And with this work, we started over 10 years ago with Dravet syndrome, which many of you know are due to haploinsufficiency for SCN1A, a sodium channel gene that encodes NAV1.1, a voltage-gated sodium channel, and its loss of function. And we started this project with two fantastic scientists at Michigan, Laurie Isom, who's the chair of pharmacology, and Miriam Meisler, who's a professor of human genetics. And Yu Lu, a research scientist in my lab, generated the iPS-derived neurons, and Luis Lopez Santiago and Yukun Yuan in Laurie's lab recorded from them. And we were expecting to find decreased sodium current because there's loss of one sodium channel. But surprisingly, compared to two patients compared to two controls, we saw increased sodium current density compared to the control in both patients, which was really unexpected. And when we looked at activity, either evoked or spontaneous activity, the patient cells had more activity than the controls. So it was kind of our first epilepsy in a dish model. And we subsequently looked at other ion channels. So early infantile epileptic encephalopathy type 13 is caused by gain-of-function mutations in SCN8A, a different voltage-gated sodium channel gene that encodes for NAV1.6. And Andrew Tidball, a talented postdoc in the lab, generated patient-derived neurons for these, gave them to Luis, and instead of a change in transient sodium current that we saw in Dravet syndrome, these patients have either increased persistent sodium current or resurgent sodium current, a delayed current activation that leads to hyperexcitability. Interestingly, different mutations have different effects on the sodium current, suggesting you might need mutation-specific treatments for these gain-of-function genetic epilepsies. We've also studied a potassium channel epilepsy using calcium imaging. We have a really cool toy called the Incucyte. So this is a microscope that goes in the incubator, and you can do regular microscopy over time, but you can also do calcium imaging. And so you can look at optical imaging of activity in the same cells over time as they form networks. And in this case, Lu differentiated these cells into neurons and plated them, and this is what the isogenic control looks like. And in this case, we didn't have a patient with KCNA2 epilepsy, so he put in a mutation with CRISPR gene editing. And you see these develop synchronous activity in networks. And when he looks at the patient cells, you can see they're different. They burst much more quickly and more irregularly. So a different type of epilepsy phenotype than we saw in the sodium channels. So we've done this type of work, other things with the 2D cultures to study potential seizure mechanisms. But we also realized, since we had the stem cells, instead of making neurons, we could use them to make other cell types, perhaps to study SUDEP. So instead of turning them into cortical neurons, we can differentiate them into cardiac myocytes, autonomic neurons, even potentially brainstem neurons to study SUDEP mechanisms. And my lab did this with Laurie Isom's lab several years ago for Dravet syndrome, because these sodium channel mutations are present in the brain and cause seizures, but they're also present in cardiac myocytes. And the idea is, the question is, are they causing arrhythmias in the heart? And so to look at that, Helen Zhang, my lab manager, generated cardiac myocytes. They start beating in culture within 8 to 10 days. And you can see the control beating nice and regularly. And the Dravet syndrome patient-derived cardiac myocytes are beating faster and more irregularly. And she confirmed that with multi-electrode array recordings, where you see the rate is more than double that of the control. And we gave them to Laurie's lab, and Luis looked at sodium currents. And similar to what we found in the brain cells, the cardiac myocytes have increased sodium current compared to the controls. And now we had 4 different patients that all showed the same phenotype. And David Auerbach, a former postdoc in Laurie's lab, did the same experiment in mice, acutely dissociated ventricular cardiac myocytes, and found increased sodium current in the Dravet mice compared to wild type. So again, loss of a channel, surprisingly, is causing increased current, and we think it's due to overexpression of a different sodium channel isoform. Interestingly, this patient, we were able to record action potentials, and it showed many after depolarizations that are the hallmarks of arrhythmia that we didn't see in the control. And the patient ended up, because of this, going to see a pediatric cardiologist, and was found to have EKG abnormalities and autonomic dysfunction. So it's kind of a way to go bench to bedside with a patient's own cells. So that's just some of the 2D work, but today, for the rest of the talk, I'm going to focus on our 3D brain organoid models, because I think this is a really exciting approach that's going to be applied much more widespread over the coming years. So I'm going to tell you 3 different stories with the organoids. One is studies of a rare epilepsy called polyhydramnios megalencephaly and symptomatic epilepsy, PMSE. It's due to mutations in the STRAT alpha gene in the mTOR pathway, so it's an mTORopathy, similar to tuberous sclerosis. Then I'm going to talk about protocadherin-19 related epilepsy, which is an X-linked epilepsy seen in females, which is really unique and interesting. And then the last part of the talk, I'm going to describe our work on understanding network activity in organoids, because after all, we're interested in epilepsy, so we want to look at neuronal function. And some of the work I was involved with in the Novich lab on sabbatical studying Rett syndrome. All right, so what are organoids? These are derived from human pluripotent stem cells. They're grown in 3 dimensions. They develop an organization that recapitulates developing human brain structures. And you can generate multiple brain regions in one organoid or pattern them to restrict them to specific regions. And the advantage over 2D, there's several. You get 3-dimensional organization, but also you can maintain them for years. Whereas with 2D cultures, after a few months, they die, they lift off the plate, you can't keep them going. So this way you can get them to really mature and form human brain networks. And they look incredibly similar to human brain in a dish. No, I'm just kidding. They don't really look like that. So basically, you start with an embryoid body derived from a human pluripotent stem cell. Many protocols use a bioreactor where they spin to keep them better oxygenated and give them nutrients. They form neuroectoderm and then an expanded neuroepithelium and then cerebral tissue. And then this is one of the first descriptions from Madeline Lancaster and Jürgen Noblick about 7 years ago now, where they make lots of different structures. So this is a stem cell zone. These are neurons in the developing cortical mantle, but they make ventricular-like structures, choroid plexus, retinal tissue. They can make hippocampal onlaga. So this is really interesting, but it's also problematic for that technique because there's so much inconsistency from batch to batch. So there are other models that have taken over and replaced that. But the organization of the organoids is very similar to an embryonic human brain. So you have the same ventricular zone with the radioglial stem cells. You have the same enlarged outer and inner subventricular zone with outer radioglia. And you have a developing cortical plate that isn't as well laminated as in vivo, but it makes deep and superficial layer neurons. Some protocols have markers of all 6 cortical layers. So there are challenges with this technique. As I mentioned, there's batch-to-batch variability. It's missing non-neural cell types. You don't have the meninges. You don't have endothelial cells. You don't have microglia. The maturation is slow compared to mouse studies. So most of them, by a few months, look like 2nd to 3rd trimester by transcriptomics. But that's not surprising because they develop on the same time course as a human brain. It takes us 9 months to develop. So you can keep them going for 9 months, a year, and get a postnatal gene expression signature. There is a limited neuronal subtype repertoire and regionalization. The original models didn't have subcortical connections. Arnold Kriegstein has some data suggesting they're enriched in glycolytic and ER stress pathways compared to the human fetal brain. So, they're not perfect models, but they're improving really fast. We're getting better and better. We can pattern them with small molecules to make specific regions, so that improves consistency. Groups are co-culturing them with human pluripotent stem cell derived microglia, endothelial cells, meninges. I'll show you one example of what we're doing. And then in terms of the necrotic center, you can cut them, or you can actually graft them in vivo and use a mouse as an incubator to vascularize your organoids as they generate. And finally, in terms of the subcortical connections, people are doing assembloids, which are combining different brain regions in organoids, which is really exciting. So one of the groups, the Park Lab at Yale, has patterned organoids into human thalamus-like structures, into human cortex, and then fused them, and he gets thalamocortical and corticothalamic connections, which obviously in the epilepsy world is going to be important for studying different epilepsy models. And then also critical are interneurons, key to many epilepsies. And as many of you know, the interneurons arise from a different progenitor region than the excitatory neurons of the cortex. So the excitatory neurons come from the dorsal ventricular zone. They migrate radially along radioglia to inhabit the different cortical layers. But the interneurons come from the ventral forebrain, the ganglionic eminences, the medial and caudal ganglionic eminences, and they migrate tangentially to integrate into the cortex. So this is an example in mouse. So the dorsal progenitors go that way, and the interneurons migrate tangentially. So they're two different populations. So to model that in organoids, Sergio Pasca at Stanford and several other groups have patterned them into excitatory cortex, inhibitory ganglionic eminence, and then fused them. And then the interneurons migrate from the interneuron organoid to the excitatory organoid to form excitatory inhibitory neuron networks to model the brain function. So these are really exciting advances that are ongoing. And I'm going to talk about some of our work with the fusion model. So we started making organoids about 5 years ago, and we started with that Lancaster and Noblick technique, but it was inconsistent. So we tried several other techniques. Many of these have multiple rosettes, so the neural rosette in there is analogous to the neural tube, but we only have one neural tube, so my postdoc Andrew made a new protocol that I'll show you with a single rosette, and these are some of the fusion techniques. We even have smiley face and cookie monster organoids. So, really, we started applying them to this disease, PMSE, thinking a disease that affects brain development and brain structure is going to be really ideal to model in the organoids. So this disorder is mainly in old order Mennonites in Pennsylvania and Ohio. There's a founder mutation, and there's a lot of consanguinity, and these kids have homozygous autosomal recessive deletions in the STRAT alpha gene, shown here, which impacts on mTOR signaling, similar to tuberous sclerosis complex. So if you lose this, just like losing TSC, you have increased mTOR activity, which leads to dysplastic brain. And basically, what's nice about this versus the TSC models is TSC is thought to have a germline mutation, but then the cells get a second hit to form cortical tubers. Here, all the cells have complete loss of function of the gene, so you don't have to worry about a second hit. And these kids are pretty devastated. They have megalencephaly, they have severe epilepsy, neurocognitive defects, and they also have areas of focal malformations and evidence of mTOR hyperactivity, shown here with increased phospho-S6. So we started this study with the spin omega technique, and this is developed by Guoli Ming and Hanjun Song when they were at Hopkins. They've since moved to Penn. But they miniaturized the bioreactor so that they could test different media combinations and come up with one that made really consistent cortical organoids. This is their spin omega bioreactor. So this was a cell paper, and actually his son, who was in high school, and two of his high school friends made that and got on the cell paper. So it was quite impressive work. So I sent my grad student, CC, down there to learn the technique, and then Louis Dang, a pediatric epileptologist and postdoc in the lab, applied it to the PMSE approach. And it was really exciting right off the bat, because when he looked at his organoids, he saw they were bigger than the control, which seemed to be modeling the megalencephaly. And indeed, when he looked more closely, this is at 2 weeks, so very early, these are the control and two patients that had this neuroepithelial budding phenotype. So they're larger, and they have these weird buds. And we looked at what could be causing them. He treated with rapamycin, which suppressed the phenotype, suggesting it was due to mTOR hyperactivity. And when he stained them with TUJ1, a neuronal marker in green, and PAX6, a stem cell marker in red, they had less neurons, but these expansions were all filled with stem cells, suggesting it was a phenotype of delayed development. So they weren't differentiating in the neurons. Instead, the stem cells were expanding. He confirmed it because he only had 2 patients and 2 controls, so he really wanted to be sure. So he made CRISPR knockout of the stratagene on an isogenic control background and was able to partially recapitulate the phenotype. So he next asked what happens as they develop further. And so now we're almost 3 months, day 85. And they actually start to look more normal. The rosettes and the neurons start to normalize. But at this time point, they express these cells that label for HOPX, that the controls have some, but not nearly as many. And I think I might have mentioned earlier these outer radial glia. that give rise to expansion of the superficial cortex. They're HOPX positive cells. And Arnold Kriegstein showed that they're a cell type that's increased in mTOR signaling. So it looks like this cell type is expanded earlier in the PMSE, and the prediction would be they would give rise to more superficial cortical neurons. So when Louis looked at nearly 4 months, day 113, he labeled for markers of deep and superficial cortical neurons. So CTIP2 is a deep layer marker, BRN2 and SAPI2 are superficial markers, and compared to the controls, the patients had more BRN2 and SAPI2, suggesting that this was leading to expansion of the superficial cortex and another potential reason for the megalencephaly. So I also mentioned that these patients had focal cortical dysplasia, so many of you know what that is. You get dyslamination, you have astrogliosis, dysmorphic neurons and glia, and this strange cell type known as balloon cells that express astrocytic neuronal and progenitor markers all at the same time. So Louis started studying this, looking at astrogliosis, and he found a marked increase, this is GFAP staining, a marked increase in astrocytes in the PMSE patient organoid compared to the control. And many of these were really enlarged, abnormal-looking astrocytes. Interestingly, Helen Badup at Berkeley has a TSC brain organoid model from human embryonic stem cells. She also sees this astrogliosis in the TSC model, so we really think it's a consistent finding. What her model has not found, and none of the animal models of focal cortical dysplasia show, are these balloon cells. So Louis looked at day 113 for balloon cells. He didn't see any in the control organoids, but he started seeing them in the patient-derived organoids. And these are really strange-looking cells. Here they're expressing GFAP, MAP2, and HOPX, so they're expressing glial, neuronal, and progenitor markers. Some of them are binucleated, which is actually classic. So if you look at some resected cortical tissue from focal cortical dysplasia, they find balloon cells with 2 nuclei, and they find expression of neuronal and glial markers that look very much like what Louis is finding in the organoid. So we think this is going to be a really nice model of human focal cortical dysplasia in a dish that we can start studying and looking at seizure mechanisms. So just to summarize this part, the PMSE patient and strata CRISPR knockout organoids show this megalencephaly and neuroepithelial budding phenotype. At later time points, they have increased outer radial glia in the subventricular zone, leading to more superficial cortical neurons. And they show these increased and abnormally enlarged astroglia as well as balloon cells. So we're excited about this FCD model. So next part of the talk, we started to ask questions, how do we improve on the brain organoid technique? And it's important to know that compared to normal development, current organoid methods start, as I mentioned, with a 3D embryoid body and end up with 3D organoids. But the normal developing nervous system starts as a 2D structure, a neural plate that rounds up to form the neural tube, and you only have one neural tube, whereas these organoids have multiple neural rosettes. So Andrew Tidball, a postdoc in the lab, decided he would go try to model the human brain development more faithfully, starting with a 2D monolayer that he patterned as excitatory cortex. He cut them into even squares and plated them in a 96-well plate, and they round up and form these consistent spheroids that have single rosettes. They have a very consistent growth rate. They grow by a micron an hour. This is over about 20 days. So by day 35, they're over 800 microns in diameter. We're doing other things with them to try to improve the technique. I mentioned co-cultures. Here, we're really excited about this. So here we've co-cultured the organoid with human meningeal cells. And if you look over time, this is in the IncuSite time-lapse imaging, these cells start diving right toward the organoid and encircling it, just like a meningeal cell layer. And the structure looks improved when the meningeal cells are there. So we think that's going to be a really nice improvement. But Andrew and Carmen Varela, a grad student in the lab, looked at the structure of these saucers, self-organizing single rosette spheroids. That's the catchy name Andrew came up with. He patterned them into dorsal excitatory cortex. At early time points, they express the appropriate stem cell markers like PAC6 and nestin. They start making neurons on the outside that express double cortin, and they, by day 42, they're making both deep and superficial layer neurons, SAPB2 and CTIP2 positive, and they have these HOPX outer radial glia. He can also pattern them to make interneurons. So they express interneuron progenitor markers like NKX2.1 and OLIG2, and then by day 56 they're making GABA and somatostatin positive interneurons. So we thought about diseases to apply this Saucer's method to, and we settled on PCDH19 related epilepsy, which is a really interesting epilepsy. It's quite unique. So it's X-linked. PCDH19 is the protocadherin 19 gene. And this is a cell-cell interaction molecule. It's got a large extracellular domain. PCDH19 is in the delta-2 non-clustered protocadherin category. And as the nervous system develops, neurons that express protocadherins interact with each other through these protocadherins and other different cadherin family members as they proliferate, migrate, and form synapses. So what's really interesting about this disease, as I mentioned, it mainly affects girls, occasionally mosaic, males are affected, but it spares male hemizygotes. So it's X-linked, so if you're male and you have the gene, it's your only copy, and if it's mutant, usually if it's a brain disorder and you're a male with an X-linked disease, you're much more severe than females. In this one, the males are unaffected, which is really curious. And the leading hypothesis is the cellular interference hypothesis. And the idea is if you have normal cells that interact to form circuits, or only mutant cells, they can still interact to form circuits as a male would. But if you're a female, because of random X inactivation, some of your cells are expressing the mutant and some of them are expressing the wild type, and these populations can't interact during brain development. So a really excellent scientist in Australia, Paul Thomas, tested this idea in a mouse model. So he made a knockout mouse that was a heterozygous female. Unfortunately, the antibodies for PCDH19 don't work well, so he had to make what's called an HA flag tag. So this is just a tag of the protein that an antibody will recognize. And the tag label is in green. If he crossed the HA flag tag with another wild type, the green is everywhere. But if he crossed it with a knockout, you see columns of green cells and columns of unlabeled cells. And these are the wild type and these are the mutant. And the mutant also express this beta-gal gene. So you get this segregation of mutant and wild type cells that can't interact. We confirmed it with a different model. So Julie Ziobro, a postdoc in the lab, who trained just down the road. She's a pediatric epileptologist who is at Children's National. She got a XGFP mouse from Sudhir Kalantri in Human Genetics. So this is just one X chromosomes labeled. She took a male, so only the labeled X could be generated from this male. And she crossed it with a PCDH19 knockout animal from Hisashi Umemori, so that the het would have one wild type allele that was green and one unlabeled mutant allele. So first she crossed it with just a wild type, and you can see the green is everywhere, it mixes in. But when she crossed it with the knockout, you see green wild type cells and unlabeled knockout cells that don't interact. So they really segregate, which is kind of interesting. So we wanted to look at the mechanism of this, and we thought the human pluripotent stem cell brain organoid models would be perfect because we could mix the cells when we make the organoids. And so Wei Nu and Sandra Mojico Perez in the lab generated CRISPR knockout female human pluripotent stem cells. They labeled different lines red and green, and they either mixed green and red wild type, green and red knockout, or green knockout and red wild type. And the hypothesis is these would form normal organoids, these would form normal organoids, but the mutant wild type combination would form these abnormal organoids with cell segregation. So what happens? So this is the wild type mixed with wild type, and you see everything mixes. This is knockout mixed with knockout, everything mixes. But when she mixed knockout with wild type, you get this pinwheel structure where the cells segregate and they don't interact, which is really, really interesting. And so to look at why this happens, She also had to, because the antibodies don't work, she had to make an HA-tagged PCDH19 that she did with CRISPR. And we know that PCDH19 interacts with NCAD-herin, and so the labeling is strongest in this apical domain down here. But when she mixed the knockout with the wild type cells start having dysregulated cellular expression of both PCDH19 and NCAD-herin, suggesting that you need the interaction to maintain the appropriate subcellular localization of these proteins as they develop. And when she looked at cortical development in the early organoids, so these are day 20 organoids, and the deep cortical layers form first, and they're on the outside. And in wild type plus wild type mixing, you get a nice laminar organization. But when she mixes wild type plus knockout, it's dispersed and disorganized. And we think this is either due to premature differentiation or a migration abnormality in these early forming deep cortical neurons. So the idea is if you have wild type, the cortex develops normally. If you have only mutant, it develops normally. But if you have mixed populations that are some wild type and some mutant, you get this altered localization of the protocadherin and ncadherin. The progenitor cells segregate, and there's laminar disorganization as the neurons form. We're also now starting to look at synaptogenesis because we think that this is critical for synaptogenesis as well. And it's possible that only the mutant cells connect with other mutant cells and wild type with wild type to form abnormal synchronous networks. That's kind of a hypothesis that we're testing. All right, so.
Focally, though, sometimes in humans, right? Very focal instead of diffuse.
Well, so there have been just a few reports of focal dysplasias in protocadherin 19 on MRI, but there's like no pathology in any of these patients. And it'd be really interesting to look at microdysgenesis, which could be more diffuse in these kids. So that's a really good question. So for up to now, I talked about structure with the brain organoids, but we want to study function because we're interested in seizures and epilepsy. So we and others are still trying to work out the best way to measure neuronal function in these organoids. We've sliced them like we would do a mouse brain to make acute slice recordings, and we see action potentials and postsynaptic currents. We've done calcium imaging. We've dissociated them and done patch clamp. We get nice action potentials. We've grown them on these multi-electrode arrays, plopping them down and looking at activity. But I want to tell you about my sabbatical at UCLA, because it wasn't just a warm winter, but I learned some really cool ways to look at function of these organoids in Ben Novich's lab, working with these talented postdocs and students, and collaborating with Payman Golshani on the calcium imaging, and Ishvan Modi, who's a really outstanding epilepsy electrophysiologist. So basically, they're making those fusion organoids that I told you about, where they pattern some of them toward inhibitory ganglionic eminence, and some of them toward excitatory cortex, and then they fuse them. So if you fuse a cortex with another cortex and label one red before you fuse it, they don't migrate. But if you fuse a cortex with the ganglionic eminence and label the ganglionic eminence red, they migrate into the cortex to form networks. And that's shown here. This is cortex plus cortex fusion, where one of them was labeled red. They don't migrate into the other side. If you fuse the cortex with the GE but label the cortex red, they don't migrate toward the interneurons, but instead, if you label the interneurons red, they migrate into the cortex. And he looked at expression of different molecules in the cortex only and GE only, and sees excitatory projection neurons in the cortex and inhibitory interneurons in the GE, so they might make the right cell types. I really like this one because I did it myself. I was at the bench on my sabbatical. And basically, what I did is I patterned this toward the ganglionic eminence, interneurons, this toward excitatory cortex. 4 weeks earlier, I labeled this red and fused them. And over the subsequent 4 weeks, these cells migrated into the cortical mantle of this excitatory organoid to integrate with the excitatory projection neurons to form a network, which is really, really cool. And Ron Mal and Samar Singh, a postdoc in the Novich lab, and Osvaldo Miranda, a technician, counted the interneurons in the cortex after fusion and forms about 20 to 30% of the total cells in the cortex are interneurons, which is not so different than the proportion in us. And they looked at synapses, and if in both cortex plus GE and cortex plus cortex, both of them have excitatory synapses, but when they looked at inhibitory synapses, they were only present when the ganglionic eminence was fused, not when two cortices were fused together, which is not surprising. But what do these do? So they looked at 2-photon calcium imaging, and this is just a control and with and without bicuculline. And you can see bicuculline is a GABA A antagonist, which should increase activity. And you see it nicely does. You get these, is it going? Yeah. You should see these blushes. Doesn't look like it's moving. There we go. So you'll see these blushes of activity when you suppress the interneurons. And the idea is you need the interneurons present to get the excitatory neurons to form normal connections and mature properly. And it's probably vice versa. The excitatory neurons help the interneurons mature. And when Ron Mal quantified the calcium imaging, if you just have cortex plus cortex, even with bicuculline, you don't see synchronous activity. But if you have cortex plus GE and you inhibit the interneurons, you get all this synchronous calcium spiking that he quantified and did cross-correlation analysis to show the increase in synchrony. So they're forming networks, at least by calcium imaging. How about by electrophysiology? And I think this is going to take us, oh, there we go. So I really like the way they're doing this in Istvan Modi's lab with the Novich lab. So basically, they use patch electrodes and they put them into the organoids, but not to patch cells, to record local field potentials. So it's like putting depth electrodes in a brain. And basically, when they compare cortex plus cortex, I'll get my pointer in a second, there we go. Cortex plus cortex, you don't get much of anything in terms of activity. But when you mix cortex plus GE and have the interneurons there, you get all these oscillations. And with this spectrogram, you can see the different frequency activity. And here's a periodogram showing delta, alpha, beta, low and high gamma activity. So it's really making oscillations just like a developing human brain would. But you need the interneurons, because without the interneurons, you don't get these. So they applied this to Rett syndrome, which many of you know is another X-linked epilepsy. It's caused by de novo loss of function mutations in the MECP2 gene on the X chromosome. And these kids start developing normally by 16 months of age. They regress. They have language disturbances, hand stereotypies, other abnormalities, and seizures in 50 to 90%. And so Novich's lab was collaborating with Bill Lowry's lab. So he reprogrammed fibroblasts from a Rett patient. And what's useful about X-linked disorders and human pluripotent stem cell reprogramming is sometimes you get clones that only express the wild type and some that only express the mutant. So he had an isogenic control to compare to. And they applied the the calcium imaging first to this. And even without bicuculline, there was more synchronous activity in the Rett compared to the control. It wasn't as much as the control with bicuculline, but it was increased, even though there wasn't a difference in the number of interneurons. They all had 25% or so. And then they looked at physiology, and compared to the isogenic control, they had these nice oscillations. There was a loss of oscillations that you see here, but all these spike discharges were present. Now what's really exciting about this technique, so here they're comparing pure control and pure Rett and doing fusions of each, but you can mix and match. And you can take a Rett cortex and mix it with a control ganglionic eminence or vice versa, and that way you can isolate whether the mutant interneurons are responsible for the phenotype or the mutant excitatory neurons or both. And so they did this for Rett syndrome. And this is just pure control and pure mutant to show the phenotype. So loss of these oscillations. But interestingly, you get these high frequency oscillations that aren't present in the control, potentially an epileptogenic marker. And this is interspike interval. So anything to the left is more frequent spikes. And notice the scale is different. This goes up to 40, this goes up to 2. So there's many more and more frequent spikes in the Rett compared to the control. So how about if you mix and match mutant cortex with control ganglionic eminence versus mutant ganglionic eminence and control cortex? Well, if you look at the periodogram, if you have the mutant ganglionic eminence, so the mutant interneurons, you lose the oscillations, except you lose the low to mid frequency oscillations, but you gain these high frequency oscillations and these spike discharges. But with the mutant cortex and control interneurons, you don't get that. So most of the phenotype is carried by the interneurons. So it's a way to kind of determine which cell types are involved in this human pathology. So I think I'll end there and just summarize some take-home points. So the brain organoids, we've used them to model PMSE, megalencephaly, and dysplastic cortex, and they implicate outer radial glial cells in the pathogenesis. The saucers, the single rosette model, is useful for identifying cell segregation phenotype in PCDH19-related epilepsy and for exploring the mechanisms. And then fusion brain organoid models support interneuron dysfunction in Rett syndrome. And I think this approach is going to be really powerful to explore these genetic epilepsies in terms of how they influence brain development, network formation, and we're just getting better and better at using the technique. So we're really excited, and I'll end there, and thank you for your attention. I just want to acknowledge real quickly, I mentioned the people in my lab doing the work as I went along, Louis with the PMSE, Yulu with the KCNA2, and Dravet, Andrew with the saucers, Shivanshi helped with the PMSE, and Helen did the cardiac myocytes. And this was collaborative work with Lori Isom and Miriam Meisler, and during my sabbatical in the Novich lab, and with Payman Golshani and Ishvan Modi. And my funding agencies are up there. So thanks for your attention.
Thank you so much. There's one very important finding that will probably shed light on all epilepsies, which is the synchronization that occurred after adding bicuculline. Can you comment on the potential mechanisms of that? And obviously, the question of how to reverse that, we know how to reverse it in 2/3 of cases, not specifically the same pathology. I mean, with medications and disconnections and what have you, but what makes neurons synchronize in that very organized manner? Also, in Rett syndrome, there was an in vivo study, I think it was a few years ago, in vivo calcium imaging that also found the synchronization of hippocampal neurons in a Rett model. And with electrical stimulation of, I think, a white matter tract, the fornix, it reduced the rate of synchronization and reduced the seizures. If you can comment on that.
Okay, so I'll take the first question. So, I mean, those are really important questions. In terms of synchronization, I think you have to consider normal synchronization and pathologic synchronization. And during early embryonic and postnatal development, there's a lot of large synchronous activity that's important for normal development of brain structures, both in the cortex and in the hippocampus, these giant depolarizations. And so if you take 2D structures, human pluripotent stem cell-derived neurons in 2D and in 3D, you start getting synchronous activity as the neurons mature. And if you add interneurons, it changes the rate of synchronous activity. I think what the bicuculline was showing was that if you don't have the interneurons and you just have the excitatory neurons, they don't make the normal small local connections that they're supposed to make, and the interneurons have to be present to make those connections, for the excitatory neurons to make the connections, and you only see them when you then block the interneurons with bicuculline. So it's kind of interesting. And there's a group, Neil Khosry in the Netherlands, looking at a lot of autism spectrum disorders and some associated with epilepsy, where he mixes excitatory and inhibitory neurons on the multi-electrode arrays and looks at the bursting activity, the synchronous bursts, and how the mutations change them, and also the proportion of interneurons changes them. So that's one issue. In terms of Rett and the hippocampus, there is in vivo evidence of abnormal oscillations in the Rett mouse models, and the interneurons are important for some of that. I don't want to say the excitatory neurons do nothing, because we see dendritic abnormalities in the excitatory neurons. But it reminded me, your question reminded me of a really exciting thing that the Ron Mullen and the Novich lab is doing. They're now patterning hippocampal organoids, and they see when they combine them with ganglionic eminence, so the interneurons migrate in, they get sharp wave ripples that you don't see in isolated hippocampal organoids. And so those are thought to be important for learning and memory, and they're oscillations that are, a sharp wave that has a ripple riding on it that are important for storing memory that interneurons fire during the ripple, and are important for setting up these sharp wave ripples. So I think it's going to be really cool to study human hippocampal development and ripple formation in the organoid models.
Thank you very much for it was fascinating and a very exciting topic and great presentation. In one short talk, you really taught us a lot about this. Question I have is, so the organoids that you mentioned, that you can actually follow them through the development of what you expect from a human organ, and follow them through the what would be postnatal kind of brain development. How far you can go through that, and then how much it falls apart in terms of like the pattern of that we expect in the normal development. And then I'm sure there are some group working on that, maybe your group are working on that, and trying to modify, with either genetic or other ways of gene modification after organization, to see if we can, how much of this can be reorganized, basically things that are disorganized can be reorganized. For example, in that mosaic pattern that you showed in the organoid. So just what was my question?
Yeah, so those are really, really excellent questions. So in terms of, my students and postdocs get tired after 4 or 5 months and give up on their organoids, but other labs have taken them out 2, 3, even 4 years is the longest I've heard. So they can survive. It's not clear that the lamination keeps improving over time. The transcriptomic signatures get more mature, but I think as we start learning what other cell types to co-culture them with, so microglia are important for synaptogenesis and are doing important things during development. The meninges do important things. So I think we're going to get better at that. In terms of the second question, I mean, those are things we want to do. Like if we treat with rapamycin in the PMSE organoids after the balloon cells form, can we reverse that? In the PCDH19 cell segregation, so I'm part of the PCDH19 Alliance Scientific Advisory Board. We meet in Rome once a year, which is really nice, but we try to come up with a treatment which is not easy, because you have to either make all the cells wild type or make all the cells mutant, and it's X-linked, and so your wild type's inactivated on the mutant cells, and your mutant's inactivated on the wild type, so there's no easy CRISPR gene editing fix or other things. But what we're trying now is to use ASOs to knock down wild type, to make everything mutant, and see if that reverses the segregation phenotype. So those are all important questions, and you have a nice in vitro model that you can study them with.
Yeah. Key.
So for those organelles, so what organoids? So what is the underlying mechanism to make the cells like form those organoids instead of like the abnormal growth, like a tumor, those kind of things? What is that? Yeah.
Yeah, that's a really good question. I mean, so there are a lot of things we don't really understand with human pluripotent stem cells. So if you don't pattern them at all, the default is not to form liver or kidney, but it's to form forebrain. We don't understand why they like to form forebrain, but that's one thing. And then they kind of follow the same temporal mechanisms of making deep layer and superficial layer. I mean, they do that in 2D rosettes as well, and we don't know what the intrinsic mechanism is that lets them know they have to do that. So those are important areas to study. All right, thank you. Appreciate it.